friends, today we are going to discuss the second topic for cheese. In our earlier discussion, we had seen a general concept of cheese. We had also seen the manufacturing process of a typical cheese as well as understood its classification and characterization. In this episode, we are basically going to deal with a specific cheese type, that is cheddar cheese. Here I have listed some of the important characteristic features of cheddar cheese. It is principally made from cow milk and its origin is from England. Now this particular cheese has a crumbly texture and it is of hard type, meaning that the moisture percentage is less than 39% here. The shape can be cylindrical or as well as in blocks, and its color is most often off-white, but a yellow-orange coloration is possible if cheese colors are added. The aging time for this cheese is approximately 3 months to as long as a year or more. Moving to the origin and history briefly here. Now, this particular variety of cheese originates from the village Cheddar, a small village in the region of Somerset in southwest England. This place is bestowed with beautiful cheddar gorge, which you can see here, which contains a number of caves along its sides. And these caves provides the ambient temperature and humidity for ripening or aging of this particular variety of cheese. And all this gives the cheese its particular name, that is cheddar cheese. You can see the caves here where the cheese are ripening. History says that Joseph Harding is known as the father of cheddar cheese making. He is credited with innovations, modernizations, standardization of cheddar cheese manufacturing process. He also took the task of distributing or making cheddar cheese common in other parts of the world other than England. Coming to ingredients which are used in cheddar cheese making. The first and the foremost obviously is milk. Traditional cheddar cheese is made with raw cow milk. Nowadays, the practices of using pasteurized or heat-treated milk. But the best variety of cheddar obviously is made from raw cow milk. The second obviously is the starter culture, which is very important in any of the fermented dairy products. Cheese, one of them. Now here the starters essentially in use are two species of Streptococcus. One you can see here, that is Streptococcus cremoris. The other is Streptococcus lactis. Now these two may be used together or as uniform cultures. Sometimes when higher temperatures are used, then the thermophilic species of Streptococcus thermophilus is also in use. The third ingredient which may be used is a cheese color called as annatto cheese color, which has a yellow-orange tinge. If applied to milk, the cheese that is produced gets a yellow-orange tinge, while mostly cheese is not involving any cheese color as such. The most important ingredient is the fourth one, that is rennet. This is the actual enzyme preparation which helps in the coagulation of milk protein casein and thus gives rise to the typical cheese that we consume. The rennet or rennet consists of the typical enzyme rennin or chymosin, which has the action on kappa casein, converting it into para kappa casein and a macropeptide. All this destabilizes the casein moiety, which gets curdled or coagulated. Rennin can be of various types. We can have a vegetable rennin, which means it is not from the calf rennin. That is, originally the rennin was derived from suckling calves, and the fourth stomach of these suckling calves were used for extracting rennin. Now this is unethical, and so various other forms like microbial rennin means rennin from microbes like fungi, and natural rennin is calf rennin, while we have a vegetable rennin here which is from plant sources. Recent day practice obviously is of using recombinant rennin, which incorporates the rennin producing gene from the calf or the cow and then inserting it into a desirable vector and producing huge quantities of the protein in cells like E. coli or yeast. Then we move on to the equipments that are required in a cheese processing plant. Very importantly, the first one used here is the cheese vat. Why is this so important? Now, a typical cheese vat can have different shapes. It can be rectangular, oval, round, but the essential feature is that it should be double-walled and mostly made of stainless steel. You can see here the double-walled nature of the cheese vat, and this hollow portion in between the double walls are made in such a way that hot or warm water can be introduced in this jacketed part so that the milk held in this vat can attain a specific temperature. The second equipment and most important is the cheese knife. You can see here a variety of cheese knives which are nothing but simply steel frames with wires, steel wires into them. Now these will help the cutting of the curd and they are known as cheese knives. After having learnt about the ingredients, we now move on to the process of cheddar cheese making. With the few slides that we have here, we will be seeing exactly how cheddar cheese is made in any cheese plant. The first obviously is milk inoculation. You can see here the milk taken in a cheese vat and that is being inoculated by the starter culture. We generally use a 0.5 to 1.5% of starter culture already mentioned, namely Streptococcus lactis and or Streptococcus cremoris. These two are mixed in the milk very nicely and then let in the same fashion for approximately 30 minutes. During this time, the milk will set into curd. After the setting of curd, which is not complete within 30 minutes, but the beginning, we generally have the milk pH being lowered, and this results in the next stage, that is renneting, means the incorporation of the enzyme rennet. This is the coagulant which will be coagulating the milk protein casein. So once the starter culture do their action, that is lowers the pH, rennin action is seen next. You can see here the introduction of rennin into the cheese vat and the mixture milk mix. This should be mixed very nicely during the rennet addition so that uniform rennin addition is achieved. After the addition of rennin, we wait for approximately again 30 minutes during which the curd sets. Now, how do a person check whether the curd has set properly or not? One of the simplest way is by inserting a blade, that is a knife blade, or a finger into the already setting curd. Two pictures are provided here. You can see the first one A, where the curd has not set because there is not a clean break. While in the second B, we can see a clean break. That means if you insert a knife or a finger, it comes out without any break in the curd and there is a glassy fracture. That gives the result that after this is completely okay for the next step. In the next step, the cutting of the curds takes place and this is again done by the cheese knives which we have already seen earlier. The next step again is cooking. Now, after the cutting action, we have the cooking part which actually implies increasing the temperature of the milk mix in the vat. How is this done? We generally introduce warm water at approximately 39 degrees centigrade in the double-walled jacketed vat, which increases the temperature of the milk curd here. Initially, the temperature is maintained at 31 degrees centigrade during inoculation, and now during the cooking step, it increases to 39 degrees centigrade. After which is the very interesting step of draining. This has been shown by various ways because various methods are employed for draining the whey that results due to the action of rennin. Rennin generally converts the milk into curd and whey. The whey should be displaced or removed, and only the curd should remain. You can see here the curds are being piled on both sides of the vat, while the center is allowed for the drainage of the whey. Now, this is a typical hand-done process, while you can see a mechanical aid being used in this second picture, where the same process of draining of whey is being done. The third one is still more interesting, where the draining is done by using certain containers to take up the curd pieces, while there is a screen on it or a stainer in the vat which removes the whey aside and keeps the curd in place. So all these three ways are meant for actually removing the whey and finally draining of the whey. So this step allows the whey to be drained, and here you can see a clean picture where all the whey has been drained from the center and the curd generally rests on the long side of the vat, both the sides of the vat. Now is the interesting step of cheddaring. All this said, this gives the actual flavor and texture. This step gives the actual flavor and texture to the cheddar cheese. The cheddaring process has several steps. Let us see one by one. In this picture, you can see those curds are being handled on both the sides and they are being stacked over each other. This is also another picture of cheddaring. The stacks are clearly visible here, and you can make the difference. This is a cheese variety where annatto cheese color has been used, while the earlier one, which is of off-white color, no cheese color has been used. The first in cheddaring process is setting. We allow the curds to settle on their own. The next step is cutting the lobes. You can see here the lobes are being cut into smaller pieces, very small pieces, and approximately of 10 centimeter, and they will be stacked on over another. See, the stacking of loaves are done here, one on top of the other. Why do we do all this? The draining of whey, the cheddaring process, all these basically releases most of the whey, because cheddar is a hard variety of cheese. Most of the whey gets released and we are left with a cheese variety, or the cheese curd here has a very little moisture. After the stacking of the loaves, that is either 2 high or 4 high, we move to the milling of the curd. There is a small milling machine, a handheld one, or it can be a larger one, which mills, that means cuts the curds into very small pieces of approximately 0.6 centimeter to 1.6 centimeter cubes. And all this is done for the salting step. If you have smaller pieces, the surface area would be more, and when you sprinkle the salt, that will be covering most of the cheese particles. This is very much wanted because salting should be uniform. You can see here the salting step, which is sprinkling of common salt on the cheese. What does this salt do actually? It has both antimicrobial action as well as gives taste to the cheese. It also eliminates any other pathogenic contaminants flora which may have been introduced during the processing. After which is the final stages, which are again not very simple. We have a packaging step here, which you can see there is a mold or a hoop onto which a cloth has been lined and the small milled cheeses are kept on top of it. Now there will be a press which will be used. You can see packaging variety again here in blocks, but mostly will have the molds in which the cheese will be pressed. So it is a hydraulic press basically, which presses the remaining moisture away and makes the cheese approximately of 39% moisture content. This is another variety or a traditional variety of cheese press which is most often also used nowadays. After which the cheese goes into aging. Now, before going into the aging, the cheeses are all dipped into hot paraffin wax. Typically for cheddar cheese, this is very essential, and they are moved into the ripening or the aging area. Over here, you can see an aging zone or a ripening zone, which should be definitely of a low temperature, around say 12 degrees centigrade or even lesser, where they can be kept for 3 months or even a year or so. But as have been already mentioned, traditional cheese making uses the caves of cheddar cheese, where the temperatures and humidity need not be maintained from outside, while that ambient temperature and humidity are provided by the caves themselves. So it's very interesting to have the difference in the cheddar cheese ripening process here. In the ripening step, actually, the proteolytic and lipolytic activities of the starter culture often plays a role in providing the final texture and aroma and flavor of the cheddar cheese variety. Once produced, after the aging, you can take it out, pack it out depending on the size and the shape that you wish to, and can procure it from the market. Now, after this, we will see a flow sheet for all that we have done. Now, actually speaking, the milk first, this is a flow sheet. The milk first is introduced into the cheese vat. Then you have fortification ingredients, which already mentioned can be colors, and starter and rennet addition are done. Finally, goes into the cheese making process. Then once the curds and whey are produced, they are separated. The curds are employed for the further cheddaring process while the whey is drained. The curds, after processing and pressing, can be into different processes, like you can have smaller pieces which are not aged, cheddar which is not aged, you can consume that also. You can go for block sizes which are aged, and you can also have very huge barrels storing the cheddar cheese which can go for reprocessing, that is production of processed cheese, which we definitely will see in our later topics. So all this said, we generally understood actually how cheddar cheese could be produced, and we had a living pictures, or you can say live picture representation of how cheddar cheese is manufactured in the industry. The cheddaring process is very, very important. It actually highlights the texture and makes it into a typical elastic or flexible cheese, while many other cheeses may be soft, while this particular cheese is flexible and it's crumbly. On storage for a long time, they generally break apart very smoothly and easily. So we have seen the production of cheddar cheese with its various equipments and certain characteristic features. After all this, we should realize that there may be certain problems during the production of cheddar cheese, and the most important one is the contamination due to bacteriophage. The starter culture that we have used often suffers phage attack. Phage means viruses. Now, these bacterial viruses often attacks the starter culture and hence loses on the functionality of the starter culture. So obviously, something should be done for this purpose. And scientists have thought about it, and researchers have done a lot of work on providing strains which are phage resistant. We can briefly look into the action, what they have done to give us phage resistant strains of the Streptococcus species, that is the starter culture for cheddar cheese production. If we see, this is actually the green colored cells are actually phage sensitive starter bacterial strain, meaning they are very frequently attacked by the bacteriophages, viruses. While here we have a lactobacillus lactis strain which are phage resistant. That means they do have certain genes which can resist the virus attack. Now, we generally go for a conjugation, a mating between these two different bacterial strains, and the phage resistant gene is transferred from the lactis strain into the phage sensitive strain. So once the conjugation is appropriate, we have the strain which now is phage resistant transconjugant cheese starter. So the genes present in these bacteria now will actually resist any phage attack, and hence we can do away with the problem of bacteriophage infection. Now, not only this, we actually encounter several problems of infection during the storage and ripening process. What happens is, this Streptococcus lactis and Streptococcus tremoris most often will die in cheese after aging for 8 to 12 weeks. And then later problem starts because the cheese plant has several lactobacillus species other than the starter culture. They generally can produce a late fermentation and gives rise to higher acidity in already produced cheese. We can also have problems like bitter tasting cheese because cheese has huge amount of protein, that is milk casein, which can be destabilized and broken down by proteolytic species present in the plant equipment. And likewise, we also have problems of molds which are frequently present on the plant equipments. Since cheese has a lower pH, the molds can easily grow and can cause certain taints or coloration in already produced cheese which is kept for ripening. So all these problems can definitely be handled with newer technology, better developments, understanding of the cheese processing and manufacturing problems and processes, and so much so, Joseph Harding, the father of cheddar cheese making, has already done wonderful work which gives us cheddar cheese which is mostly free of any spoilage and contaminants. Thanks to him, and thanks to all of you for your patient hearing and listening. We can all enjoy a cheddar cheese, which is the favorite cheese for all of us all over the world. The hard variety of cheese available in any of the supermarket or even the common shop near to your place. And that is why the importance and popularity of cheddar cheese. Thank you.